And really, already, so to say, the whole reconstruction of the C. elegans worm, its neuronal network, was the first example of really volume EM. But it's been a bit unnoticed, and there's been lots of developments going on, and that's why we call it a bit the quiet revolution as compared to the resolution revolution, a bit of rivalry maybe. And so it's been going on for a long time, but especially in the last decade, the developments in technology have taken a right pace and transformed the way what we can do actually with volume EM. So volume EM is really a collection of technologies. So it's not a single instrument, like for instance a cryo-EM would be, but it's a collection of instruments. It can be a transmission electron microscope, but it can also be a scanning electron microscope. With the scanning electron microscope, we have different technologies. We can do array tomography, where we collect sections on a wafer or on a grid and then image those. We can do signal block phase, SEM, scanning electron microscopy, and I'll highlight an example of that. Or for instance, with a focused ion beam, we can mill away material and then image that new surface. And I'll show one example of that as well. And I will start off with an example where we've done serial TEM tomography, but I'll come back to that in a second. When we talk about volume EM, we talk about a volume, of course, so 3D structures, and usually we talk about scales that are a micrometer or even bigger in volume, nanometer resolution. And that's that real challenge of combining the resolution with a large volume. What we're looking at is organelles and larger structures. So organelles, cells, cell interactions, and those kinds of things. Sometimes we say a volume has to be thicker than 1 micrometer. That's not a fixed measure, but that's a way of so to say delineating it as well from, for instance, a single electron tomogram or cryo EM. We are in this case, of course, when you do single cryo EM, you're also building a 3D structure. And in principle, that has a 3D volume. That is not what we are talking about. We are not talking about molecular structure, but really anything up. And in most cases, we are talking about plastic resin embedded material, where at the moment we haven't included cryo-EM as such in our initiative. But there are lots of developments taking place that will certainly give cryo-EM a place within the volume EM community. Now, we have this selection of technologies, but each has its own advantages, but also disadvantages. Some are fast, some are slow, some can deal with a very large volume, others can deal with a higher resolution. And so it's always, again, the biological question that drives what kind of VEM approach you should take. So I've highlighted the connectomics of the worm, C. elegans. They've done that with serial section TEM. Nowadays, we would not do that anymore. And I'll try to explain why we would not do that nowadays anymore. So what I'll start off with is give a few examples of work we have done, and I'll explain then later why I'm showing the things we have done. And I'll talk about cell biology, that's my own background, science, and antimicrobial resistance, and the different technologies we've used for that. As Johanna, I'm a cell biologist, and I look a lot at intracellular trafficking. We do a lot of correlative microscopy. Indeed, that's really what also is my background. And so a long time ago already, we are looking at the endocytosis of material from the outside. So if we take, for instance, the EGF receptor here, that's internalized via clefrin-coated pits, ends up in the early endosome, and then that signal the EGF, because it's telling the cell to divide, that needs to be degraded. At the same time, the transferrin receptor, which binds transferrin, so iron uptake for the cell, that's also taken up via that same structure, although there's debate about that, and also ends up in that early endosome. But that receptor can be reused, so that is recycled back to the plasma membrane. Now, that segregation mechanism here in the early endosome is something I'm interested in. And so what we did is we made a transferrin that was tagged with a red fluorophore, and in addition, with a 5 nanometer gold particle. This means that we can see the transferrin both in the fluorescence light microscope, but also with the 5 nanometer gold particles in the electron microscope. We've done a similar thing for the EGF, the epidermal growth factor, but in this case, we tagged it with a green fluorophore and a 10 nanometer gold particle. So now we can discriminate those two probes, both by color and by size. And what we've done is we've internalized that into the cells. In this case, these are standard HeLa cells. And I do apologize for the light microscopists in the audience. This is not the best microscopy you can get for fluorescence, but there's a few things that are going on. This is with very limited amounts of the probe there, so that we don't oversaturate the system. But what we're looking at, so this is one cell, the outline of one cell, and I want to draw your attention to this area, because here this is again sped up about 5 to 10 times, and you see the movement of this structure, which is an endosome. It contains both red and green because it's yellow, and at a certain point you will see a green emerge there at this, and then the cycle starts again, the movie starts again, because at that point we fixed that moment in time, and we wanted to know, okay, what's going on at that point, and that's highlighted here. So we have minus 15 seconds, minus 10, minus 5. This is our last frame of the image. We then still have about 3 seconds before we really have the image that that we can see in the electron microscope. We trace back that same position we have here in the electron microscope, and that's all about correlative microscopy, which I'm not going to talk about. And so we see this structure, we overlay that, and then zoom in. And this is our 2D image structure. So with that, the red arrows point to the small gold particles. The green ones point to the larger ones. And they, although this is one yellow blob with a bit of green and a bit of red, the segregation at the electron microscopy level is much, much higher. There seems to be a connection here as well. And who knows what's going on here? And that's the problem with a 2D image. We cannot see the whole overall structure. So what we decided to do is we made serial fixed sections, and we reconstructed that volume. And so this is first, second, and see the blips going every time, and that's a new section which has been reconstructed. Then we've modeled that, and we see that this larger structure that is now connected with this red structure, but also with this structure. And I'll let it play once more, because I want to draw your attention to this purple pink structure. And I'll let it play again, because that's where the volume EM aspect of this project came in. Because what we noticed is that when we reconstructed this volume, there is an extra structure which we hadn't noticed in the 2D image is present. And that is exactly that connection between this endosome and the plasma membrane. And that's exactly what we were looking for. Those kinds of connections, recycling of transferrin. And so only by doing this serial section electron tomography could we find that connection. So that's where it was really important, and we wouldn't have found it any other way in this case, to find that connection between the endosome and the plasma membrane. The next project about is even more challenging in that sense, because we're looking at single cells inside a brain. And this is really the needle in the haystack problem. How do you trace that? So we want to see a specific neuron inside the brain. So this is a project done by Rob Lees, and he'll be he's now in Oxford, and so we've loaded one specific neuron with a fluorescent dye, and then we're going to try to retrace that same neuron inside a large volume at high resolution in the electron microscope. So what we do with that is, because it's impossible to find really that one cell if you would just go sectioning or even turn on a serial block phase SEM or a FIB-SEM to retrace that. So Rob used a trick here, and that's based on work mainly done by Graham Knott already a long time ago, because we're using the power of, in this case, a multiphoton microscope, which we use for deep imaging inside the brain, but it's a very powerful laser, and we can use that laser to make branding marks. So what we do, we have our neuron of interest, and then we basically burn marks inside that neuron of interest. What we do then is we go up a bit and we make a larger structure around it, burn another structure in there, and then we go up. We need to be to analyze that volume. Normally you would put an X marks the spot. That, of course, in this case would not have been a good idea, because then we would have burned away our neuron of interest. So what do we do next? Well, this shows, so to say, our guiding system. These are these brand marks, which are nicely in the fluorescence microscope as well. And this is still our neuron of interest. That's what we want to look at. What we're going to use is then, in this case, a serial block phase SEM, because the neurons, well, some neurons are almost a meter long if you look at the peripheral nerves in your leg in a mouse brain, but still the length of that neuron is limiting, for instance, for something like serial section transmission electron microscopy. So we're going to use serial block phase SEM in this case, and I'm going to show you a movie, and it's sped up. It's about 5 times faster than what it was meant to be, and this is from Zeiss with a Gatan Freeview system in there. What it has is they've built a microtome inside the scanning electron microscope. It scans the top surface, then slices off that top and scans it again. And that's repeated over and over again. And by that, you can build and collect hundreds, thousands of sections if you want, and build up a real 3D structure. And so that's what we did for our structure as well. So we've now zoomed into the area where our neuron of interest is, and then we're going to look at that. And this is really the whole volume of what has been taken. So you can see all the ultrastructural details. So we have the resolution, because we want to look also at connectivity between those neurons. And then again, we segment that volume and draw in the neurons of interest. And in this case, we are interested in this green neuron, looking at the interactions with other neurons. And this is a large volume. This is microns, several microns long. And would only have been possible, again, by directing us to the right place to look, but also because we have this serial block phase SEM, we could collect a large volume at the resolution we need. This, like I said, this was done by Rob Lees and developed that whole protocol of doing that, really retracing a single neuron inside a brain. So this is where, in this case, we had to use serial block phase SEM, because the TEM, it just would have taken us months and months of work to reconstruct that volume. Whereas the serial block phase, it's automated. You can collect that overnight, over the weekend. And so it's much easier and collects things at the resolution. It usually sections at about 50 nanometers thickness. So you do collect 2D images that have a certain depth. And so your overall resolution is limited in that sense, but we are looking at connectivity. So that's where our 50 nanometer, so the Z resolution, is good enough. The last part I'm going to talk about is antimicrobial resistance. Now, we all know that antimicrobial resistance is really a big issue. And it's also an issue in dental care and research. And that's where we work together with Josh Jenkins, who was a PhD student in the School of Dentistry, in the group of Professor Bosu. And it's one of the, I think, one of the most interesting projects we have done, or one of the clever projects, I found it. So rather than using chemicals to kill bacteria, for instance, they were trying to look for a physical way of killing bacteria. And so what they created is a surface, titanium, and then etched away of a lot of that so that it creates nanospikes. And this is an SEM image of those nanospikes. And then by putting on, for instance, E. coli in this case, but it works for other microorganisms as well, putting those on those nanospikes, we're trying to rupture those and kill them. And this is what looks like an SEM image. This is what looks like a deflated bacterium that might have been punctured. But that's what we want to know. Is it really punctured, or is it just coincidence that we find that deflated bacterium there? And so that's where, how do we access that particular structure? Embedding this in a standard way for TEM and then sectioning with a diamond knife is not an option. These are titanium nanospikes, so they would immediately kill your diamond knife. So TEM, but also serial block phase SEM, would not be an option to look at that. So that's where we went to focused ion beam SEM. That's really where you use the power of a gallium ion beam, for instance, in this case, to mill away part of your material so that you have a smooth surface again that can be analyzed using the SEM. You scan that surface, and then you mill away a very tiny layer. And that's the good thing about the FIPSEM. That has a much, so to say, shallower depth at which it can mill away. So that can mill away 2 nanometers, 4 nanometers, depending on your needs. And then scan that again. So your overall Z resolution is much higher than you would get for a serial block phase SEM. But you can also use it for preparation. And that's the other things we did is we used it to mill away most of the material behind and before an object of interest, and then did a lift out. Now, this is all done at room temperature, so it's not as difficult as, for instance, it would be making a cryo lamella. But we tried both. We used both ways, both the lamella lift out, but also the direct focused ion beam image SEM imaging we used for this project. Really, this is the top view SEM, and then we are going to mill in this area and find really, this is one of the needles. The second one is here, which are depicted here. And then we're going to look at that structure. And this is then what that looks like. So, yep. 1 nanowire is not really penetrating. This one we think is penetrating. But again, that's where at that point we were a bit limited with the FIP imaging itself. And that's where we did the lamella lift out, and that's where we really could see that some of those nanospikes are really penetrating the bacteria. And like I said, that also works for Staphylococcus aureus. That really is where we use the FIP-SEM to really analyze that sample, and it was the only way we had to use FIP-SEM as that sample, because none of the other methods would have been possible. So that's nice. What's the problem? Well, there's the big problem here, because when we did those projects, we did the first one we did in-house. That was in Bristol on our 200 kV TEM equipped with electron tomography, so that we could easily do. But still, not every facility will have a 200 kV TEM. We didn't have a serial block phase SEM at that time, so we collaborated with Lucy Collinson at the CRIC and used their serial block phase SEM. So we did all the preparation, we did the infrared branding, then took that sample to the CRIC and did the SEM analysis. The last one, where we used this focused ion beam, we didn't have any access to a focused ion beam SEM, so we had to go to the School of Materials, in this case, in Manchester, to have our sample analyzed. And in a sense, we were lucky because we could access all those instruments, because we know Lucy very well, we had good relationships with Manchester, and so we could get access to that. But there's no system how you could get other equipment. And that's really what started for me, the VEM community exercise. That's stuff. As after we published a paper with Lucy on the serial block phase SEM, that's where we started discussing how can we make volume EM technology better available for the whole community. And together with Gerard Kluivert and Ardan, we organized the first town hall meeting. And if you look at Gerard and Ardan, hopefully you'll recognize that they were mentioned in the announcement as well. This is EMBL EBI. So 2 people out of the 4 that really started this are data driven. And that's one of the things. I'm not going to talk about data today at all, but a large part of volume EM is data analysis, data storage, data annotation. And so that's why they are heavily involved in this whole process. We were kindly hosted by the Wellcome Trust at that point. So we in person, a rarity nowadays, at the Wellcome Trust, where approximately 60 people joined there, and we discussed, okay, what is the state of the art of the technology? Where are the gaps? Where are we going? What does the community need? And that's where we developed a kind of a plan to get infrastructure. And this is at the moment, at that point, it was still very UK driven, where we started our building of the community, first in the UK, but later on internationally as well. Then in 2020, we had our second meeting. Now it's Zoom times, and we still had about 60 participants, but that really focused our minds on, so it's nice to have those dreams about we need an infrastructure. how do we get there? What do we need to do as a community to make that possible? And that's where we organized ourselves. And again, I should emphasize, this is driven by all the researchers that are involved. This has no funding yet, as I'll come to as well. And it's really community driven and all based on volunteers. But that's where we organized ourselves into a number of working groups, 6 working groups in this case. And what are we to do in that case? Well, initially we were trying to build capacity to meet the VEM demands. Initially for the UK bioscience, but more and more this is not a UK problem. This is a European, worldwide problem. So this is now, you can almost cross out the UK and make it into a worldwide bioscience research community. What do we need to do in order to do that? We need to build that community and come up with plans and use each other's expertise and things like that to really drive and in that way drive to see that we can build that capacity. So it's an ongoing cycle where we benefit from each other and try to influence also the funders in that. And so the 6 working groups that were formed are the following. And as I said, this is all volunteer based. There's no funding associated with it yet. Also, if you look at this, so these are the chairs of those different working groups. So community, outreach, sample prep, data, training, and infrastructure. And I will go through each in a little bit just to discuss what they are doing. So community and outreach look a bit similar, but community is an inward exercise. This is really keeping all the VEM experts up to date, to have them talking to each other. What are the latest developments? How can we learn? The outreach really to go out into the bigger scientific community and talk about, this is what we can do with VEM. Have you thought about using VEM? This, if you want to look at a larger volume, this is now the new technology. This is what we should be doing. Training is of course is a very important exercise. We all want to make sure that the expertise we have is not lost when, for instance, we move on, we retire, or also we want to train the new young scientists in that area. Data I've talked about is one of the key things for a VEM experiment. But before that, sample prep. VEM has, especially the serial block phase SEM, needs a different kind of sample preparation as other EM technologies. So that needs, again, special protocols. Can we make a standardized protocol with 100 electron microscopists? You will probably have 101 protocols as well. So can we make sense of that? What works and what doesn't work? And then the last part was still that infrastructure. So how can we, because we know that there's a need for more instruments and especially also for more staff to run those instruments. And initially that was UK focused, but more and more we are already thinking about, how can we organize that in an international setting? So those are the brief things the working groups try to do. So what have they done? Well, the community is really, we have a number of Twitters. You can use and join us. That's I think one of the messages from today's presentation. If you're interested, join us, and you don't need to write down anything or take a screenshot at the moment. Everything will be available after the presentation. So we do have a kind of a website. It's in development, and I'll highlight that later as well. Outreach is really or the group basically has developed a number of surveys to also see what's available in the community already, what is missing, and especially also develop case studies just to highlight to people that are not familiar with VEM that actually some of the key scientific findings are based on volume EM technology, but because it's still silent community, or it's a silent revolution, we don't make too much fuss about it. But there's so much going on, and we need to highlight that much better. So this is one of the surveys. Again, you can take a screenshot to look at those, but everything will be available. Infrastructure, we are still working, and we're trying to keep the discussion going with, in this case, the UK funding bodies, more and more looking at an international perspective as well. Data, again, there's a survey on data, how we use it, how easily is it to use the things. Sample prep, that's again where the group is really trying to develop and discuss what are the best approaches for sample preparation in a VEM setting. And of course, last is but definitely not least, there is a VEM training survey. Is there any training in volume EM? We don't know what is available. What do we need to develop as a community? Because we should be providing that training to new users as well. So that is up until the end of 2021, when we had our third town hall meeting to discuss the of this initiative. And then all of a sudden, also maybe because it was still Zoom times, we went from about 60 participants to 250 participants. And it went much more international. That's really where we reached one of our objectives, is really building that community, still growing. And so that's where we want your involvement. And as I said, all that documentation is openly available at Google Drive. I'm almost done, and I'll finish off with one of the successes we've recently got is that with a group of researchers, we applied to organize a Gordon Research Conference, one of the most prestigious conferences probably in the scientific community. And we were awarded that the opportunity to organize that first Gordon Research Conference on volume EM. And I'm very happy to announce that will take place next year, July, in California, USA. And keep on trying to build that community further. And so there, I would like to finish. As I said, there will be a lot of names, but there's many more, I think, now already involved in that whole process. And with that, I would like to finish, and thank you for your attention, and happy to take any questions.